Hi, I'm Hrishi, and my partner is filming, and our project is on our poster is on the history of bacterial transformation. So, bacterial transformation, there's a variety of mechanisms that bacteria use. The three main important ones are transformation, conjunction, and transduction. And so each of these this one, transformation was discovered in 1940, in the 1940s. Conjunction was discovered in 1946, and transduction was discovered in 1951. So the way they discovered plasmids was through bacterial conjunction, and they discovered plasmids in 1952, and it was determined circular DNA in 1962. So restriction enzymes and plasmids help recombine the DNA. That's the whole way that bacteria transform themselves using plasmids. And plasmids, they're actually extra chromosomal DNA, and this is what is transferred in conjugation, that's the diagram of it. So doing conjunction, or why did I say conjugation? During conjunction, the bacteria form a bridge between each other, and the plasmid is what is transferred through that. Transduction is usually through bacteriophages when the virus eats DNA, and then it just somehow transfers that DNA to some other bacteria. And then transformation is when, like, some dead DNA is encapsulated into a live DNA. So then here's our quiz. So when were restriction enzymes found? And restriction enzymes were found in 1960. I'll tell you after this. This is wrong. This is the first draft. And so this is the conjugation bridge. So this is bacteria 1, this is bacteria 2. So this is the F plus gene and the F minus gene, and the F plus gene codes for these little hairs, the cilia on the bacteria. And using conjugation, it can transfer this F plus gene to the bacteria 2, so then it'll start growing hairs and cilia. That's it. No.
Alright, so most plasmids are extra chromosomal circles of DNA that could replicate by themselves in the cytosol of bacteria. To copy themselves in the cytosol, plasmids must have a starting point for replication, which is called the origin of replication. The origin has recognition sites for DNA polymerases that replicate the DNA and enable the plasmid to be cloned as the bacterium divides. Plasmids also contain genes, and this is the GPLO plasmid map. The plasmid map shows the location of the origin of replication and the GFP, BLA, and ARA-C genes. A plasmid must have a promoter upstream of the coding region of the gene. Genes of interest are ligated into cloning or expression plasmids to make more copies of the gene or to express proteins. Recombinant DNA can be used in detergents and as pharmaceutical drugs. Clone genes can be used to make genetically modified crops and for gene therapy trials.